So we mentioned that in TCGA studies, a lot of the well-known recurrent mutated genes are discovered. The exception is actually epigenetic regulators, because previously people, when they look at the expression, sometimes EZH2 is an epigenetic factor. It is also highly expressed, but that's not always the case. A lot of these epigenetic regulators, they might only be slightly overexpressed or lower expressed. The differential expression doesn't give you a very strong signal. Also, copy numbers sometimes also don't give you a very strong signal. It's only when you look at the mutations, you see this particular mutation happening. And some of them can happen in, you can see here, the gain of function and loss of function in quite a number of patients, could be as high as like 80% of some patients. And suddenly people say, wow, that's something, right? It's really cool. Especially if you see these, really dark red mutations that are gain of function mutation in specific cancer at say 80% of the chance or the cases, that's when people are very interested in developing a drug that's really specifically inhibit that mutation. Hopefully that can be used to cure the cancer. So about 10 years ago, around the early days of tumor sequencing, many companies started investing in epigenetic drugs, and they're still working on this, and some of them are also already in the clinic. In terms of DNA methylation, you can also see that, for example, with DNA methylation, they see, for example, in this colon cancer type, there's one group of genes that are having very, very different methylation profile in a subset of patients. And they call these a CPG island methylation phenotype. And if you look at so by the way, the columns here are the samples and the rows are the genes, right? And you can see here, this group of samples, their methylation pattern, at least on some of these genes are very, very different. When you look at whether they are genetically different, interestingly, you also see a BRAF mutation. Somehow when the BRAF gene is mutated in colon cancer, these genes, many group of genes, their promoter methylation suddenly change. This is another example for glioblastoma. You can see here, this group of samples, their DNA methylation pattern. By the way, these are from different TCGA studies. Every tumor type, they publish one study. This is in the glioblastoma, they call it glioblastoma CPG island methylation phenotype. There's a subset of patients with very different genes, epigenetic profile. And interestingly, this is associated with another IDH1 mutation. By the way, the scientist who discovered this IDH mutation is a Dana-Farber Bill Kaelin, who won the Nobel Prize last year for this kind of discovery. It's a epigenetics mutation, which is associated with oxygen sensing. And then in gastric cancer, again, you see a subset of these patients. Again, they are associated with viral infection. And if their tumor is caused by the EBV virus, they have very different DNA methylation patterns. And you can see here the EBV infection. And finally, there's in kidney cancer, another group of patients, very different DNA methylation patterns. I guess they still don't. Also, this is another mutation with a gene. So you can see sometimes a particular mutation on the gene is associated with an epigenetic pattern that you see. Yeah, so you can see here these two from both the colon cancer and gastric cancer, the orange group, not only they have mutations or viral infection, they could also have macrosatellite instability. These are repeats in the genome that suddenly become activated. And you will see associated epigenetic changes and also associated, you can actually see this also from gene expression as well. And for the other group, in both IDH mutation and fumarate hydroxylase mutation is related to the energy metabolism. And in both cases, they have very different survival. So for example, with IDH mutation, yes, it happens, but when the patient has this mutation, they respond better to treatment. Whereas for kidney cancer, when they have this particular mutation, the epigenetic status, different metabolism, they have much, much worse survival. This is because in general, I think kidney cancer have better survival, and generally glioblastoma has very different, very, very worse survival. And so when they are caused by the same or similar mechanism, one is actually better, the other is worse. That's compared to the norm of those cancer types. Okay, so you can see epigenetic mutations happens a lot, and also they could be associated with other mutations. It could be associated with metabolism, or associated with survival, and that's why there are more drugs developed against those. There are also drugs targeting DNA methylation to make the tumors less DNA methylated, and we'll show you some of that in the cancer immunology lecture.